So, good evening, everyone. Good evening, sir. I am Muhamim Minga-as, and we are the group 1. So, today we are going to discuss the nucleic acid metabolism. So, what is nucleic acid metabolism? It involves the synthesis and degradation of nucleic acids, DNA and RNA, within a cell. So, it includes the processes such as DNA replication, transcription, and translation. These processes are crucial for cell growth, development, and the maintenance of genetic information. So, additionally, nucleic acid metabolism encompasses repair mechanisms to correct errors or damage in DNA, ensuring the integrity of the genetic material. So, just like what I've said, the nucleic acid metabolism encompasses the formation and polymerization of nucleotides into more macromolecular structures such as DNA and RNA. So the further modification of these macromoleculars and their catabolism and excretion. So what will happen if our nucleic acid metabolism is not present in our body? First, we have the genetic disorders. So genetic disorders occur when a mutation uh, affects your genes or when you have the wrong amount of genetic material. So genes are made of DNA, which contains instructions for a cell functioning and the characteristics that makes you unique. So I mentioned the DNA and RNA. So today we are going to differentiate the both. First, we have the DNA. So DNA is a deoxyribonucleic acid. It stores and transmits genetic information. It is a double-stranded molecule, look like a spiral staircase or twisted rope ladder, just like what you've seen in that picture below. So first we have the DNA structure. So the DNA structure, double helical nature, discovered in 1953 by Watson and Crick. So DNA is made of two strands of nucleotides that wraps around each other to form a double or spiral double helix. So in RNA usually exists as a single-stranded molecule basis. So sugar type DNA contains a deoxyribose sugar. So what is deoxyribose sugar? So deoxyribose sugar is a lack of uh, atom or molecule in one substance. So RNA contains ribose sugar structure. So next, we will have the bases. So DNA contains adenine, thymine, cytosine, and guanine. In RNA, we have the adenine, uracil, cytosine, and guanine. So the RNA replaces thymine with uracil. So the function, what is the function of the DNA? So it stores genetic information and serves as a template for RNA synthesis. So RNA plays a key role in protein synthesis, or mRNA carries amino acids to ribosomes or tRNA and helps in various cellular functions. So the location of DNA, so it is usually found in the cell nucleolus and eukaryotes. So RNA can be found in the nucleus or pre-mRNA and cytoplasm. So DNA, it replicates during cell division to pass genetic information to daughter cells. So RNA, it generally synthesized as needed and doesn't undergo the same replicating process as DNA. So, in summary of total, no, DNA is the primary genetic material storing information for cellular activities. Meanwhile, in RNA, it acts as a messenger and participant in protein synthesis, among other functions. So next, we'll have the DNA structure. Just like what I've said, DNA is made of nucleotide subunits. So there are three parts of DNA nucleotide. First, we have the deoxyribose, which is I've explained kaganina. The deoxyribose is a ribose that is lack of one atom or molecule. Second, we have the phosphate group. So third, we have the nitrogenous bases or the ATGC. So we'll move on onto the next slides.
So, nitrogenous bases in DNA. So, nitrogenous bases are molecules that contains nitrogen and a chemical property of a base. So, the nitrogenous bases in DNA are adenine, thymine, guanine, and cytosine. So, adenine pairs with thymine, while guanine pairs with cytosine. The pairing occurs because the complementary bases form hydrogen bonds with each other. Adenine and guanine are purines. These are two-ring bases, while thymine and cytosine are pyrimidines. They are one-ring bases. So adenine and thymine form two hydrogen bonds, and cytosine and guanine form three hydrogen bonds. Next slide, please. So the nitrogenous bases makes up the rungs of the DNA ladder, and the bases are held together by hydrogen bonds.
So for DNA replication, it is the process of making a copy of the DNA. So the DNA replication or the DNA synthesis is the process by which the genome's DNA is copied in cells. So before ang cell mag-divide, it must first copy or replicate its entire genome so that each resulting daughter cells ends up with its own complete genome. So genome is the complete set of DNA or genetic material in an organism. And the purpose of DNA replication is to produce two identical copies of a DNA molecule. This is essential for cell division during growth or repair of damaged tissues. And the DNA replication ensures that each cell receives its own copy of the DNA. And there are enzymes nga na-involve aning DNA replication. Kani nga mga enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So first is the helicase or the unzipping enzymes. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. Second is the DNA polymerase or the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Third is the primase, which is made of RNA, is the initializer. With as great as DNA polymerase is, ang DNA polymerase, dili siya maka-figure out kung asa mag-start without something called a primer. So the primase makes the primer so that DNA polymerase can figure out where to go to start to work. And ligase, or the gluer, it helps glue DNA fragments together. And next slide, please. The steps of DNA replication. So, first is the helicase enzymes, which is katong the unzipping enzymes. It separates two strands of nucleotides, or it breaks the hydrogen bonds that holds bases together. The point at which the two strands of DNA separate is called the replication fork. Next. Second is the another enzyme, which is the DNA polymerase, or the builder, binds to separated strands and starts moving along original DNA. And ang DNA polymerase, mag-assemble siya og a complementary strand from free nucleotides that are found in the nucleoplasm. Next. And the nucleotides are joined to new chain by covalent bonds by the phosphate group and the deoxyribose sugar, and are joined to original DNA strand by hydrogen bonds. For example, the original DNA sequence is ATTCCG. So the DNA polymerase is magbuild siya og bago nga strand that is complementary, or TAAGGC. And in case you don't understand the process, I will be showing you a very short clip to further discuss this kind of process.
DNA. We talk about it so much, it's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, It makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors. In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions in the video details to explore more.
So I hope all of you understand the short video clip that was being presented, and the summary of replication is that ang replication dili siya magbegin on one end and move to the other. Instead, mag-start siya and mag-occur simultaneously at different sites along DNA molecules. For example, the replication occurs at 6,000 different sites in fruit fly DNA, and it allows for faster replication. And the end of replication is duha ka identical DNA molecules, each consisting of one new strand and one from original DNA molecules. So, next slide, please.
So among some kaning nucleotides, minor lang ang magkaroon ng error. So errors in DNA are called mutations. So a mutation is a change in the DNA sequence of an organism. These changes in DNA sequence can have serious effects. Enzymes proofread nucleotide sequence, reducing error rate to 1 per 1 billion nucleotides. So mutations can result from errors in DNA replication during cell division, exposure to viral infections, or radiation. Next slide. So other agents that damage the DNA are chemicals and radiation. These can lead to mutation that could result in cancer. So harmful mutations may cause genetic disorder or cancer. It changes all aspects of its life because mutations are essential in evolution. Without mutation, evolution could not occur.
So now let's talk about the nucleic acid. So RNA is for moving genetic info from DNA in the nucleus to the site of protein synthesis in cytosol. So, ang RNA is a short life, constantly being made and destroyed. RNA molecules comes from a genetic information, and RNA not only carries information, it also puts that information to work by directing operation in our cell. So each RNA molecule uses its unique combination of letters to make our cell to all sorts of useful important things like produce proteins or catalyze chemical reactions. Next please. So RNA have three parts, RNA nucleotide. So the first one is ribose or sugar. It's alternate with phosphate groups to form the backbone of the RNA polymer and binds to nitrogenous bases. The true phosphate group. The phosphate group is important na siya in activation ay activating proteins para ang proteins can perform particular function in cells. Then nitrogenous base, the last one, also called nucleobase because they play major role as building blocks of the nucleic acid in the DNA and RNA. Next, please. So the difference between DNA and RNA. RNA is a single-stranded while DNA is double-stranded as sa discuss ni Muhamim Gaina kung nakakuan man. So, and RNA contains uracil while DNA contains thymine. RNA has the in ribose while DNA has the sugar deoxyribose. Next please. The types of RNA. So the first one is messenger RNA or mRNA. So single uncut strand of RNA. So mRNA instructs cells in the body to make specific proteins, and proteins play many essential roles sa tong lawas. Ang RNA basically uses our own body and biological process to potentially treat diseases and prevent infection. Number two, second one is transfer RNA. The primary function aning tRNA is to bind to a specific mRNA codon and then transfer an amino acid chain being created in the ribosome. The last one is next, please. The last one is ribosomal RNA or rRNA. Kanisha is responsible for translation or the process our cells use to make proteins. RNA is responsible siya for reading the order of amino acid and linking amino acid together. Next, please. So, Miss Madam.
Okay, so transcription. Transcription is the first step in gene expression. Transcription is performed by enzymes called the RNA polymerase, which link nucleotides to form RNA strand using the DNA strand as the template. Transcription information is rewritten. Transcription is something we do in our daily lives and is also something our cells must do. So, in biological term, So there are three stages or steps of transcription. We have the initiation, the linguation, the second stage, which is the elongation, and the third stage, the termination. So for the first step, the initiation, we have RNA polymerase binding to specific region of DNA called a promoter. So promoter is the beginning of the DNA strand that signals the start of the RNA transcription. So once the RNA polymerase bonds to the promoter, the DNA will separate and one strand of DNA will be used for transcription. Next slide, please. So we can see the figure here, the initiation stage of transcription. So the RNA polymerase binds to the region of the DNA which is the promoter. So once the RNA polymerase binds to the DNA, it divides the DNA into two parts, which which provides us the single-stranded template and the which is the below part and the upper part is the coding template. Next slide, please. So the second step or stage of transcription, which is the elongation. So RNA polymerase bonds to first the DNA nucleotide of template strand, then begins adding complementary RNA nucleotides to newly forming RNA molecules, following complementary base pairing rules except uracil pairs with adenine. Next slide, please. So in the figure, the template strand acts as the template for RNA polymerase. So the RNA polymerase reads the template one base at a time. So it one base at a time means reading the or pairing the adenine to thymine and uracil to the to adenine. Then the polymerase builds an RNA molecule out of complementary nucleotides, making a chain that grows from 5' to 3'. So the RNA transcript carries the same information as the non-template or the coding strand. But the only difference is that the RNA polymerase contains uracil instead of thymine. Next slide, please. Okay, so moving on to the third stage of transcription, which is the termination. So the RNA polymerase keeps adding or continuously adds nucleotides to RNA strand until it reaches a DNA region called the termination signal. So in initiation stage, we have the promoter, which signals the start of the RNA transcription, while in the termination state, we have the termination signal, which signals the stop of the RNA transcription. So once termination signal is reached, RNA polymerase releases DNA and newly formed RNA molecules. Next slide, please. So in this figure, we can see that the terminators signal the RNA transcription to stop, which shows the completion of the RNA transcript. So once they are transcripted, this causes the transcript to be released from the RNA polymerase, which is the red line. Next slide. So moving on, DNA strands will rejoin and the mRNA will leave this nucleus via a nuclear pore and head to the cytoplasm where it can direct protein synthesis. All three types of RNA, which are the mRNA, tRNA, and the ribosomal RNA, they are made via transcription and all three will play a role in protein synthesis. Okay, next slide.
So, protein synthesis. Proteins are made of amino acid subunits linked together by covalent peptide bonds. Protein synthesis is the intricate molecular process through which cells construct proteins, the fundamental building of blocks of life. This intricate and highly regulated mechanism involves the transcription of genetic information encoded in DNA into RNA, followed by the translation of RNA into specific sequences of amino acids that ultimately form proteins. This sophisticated biological dance occurs within the cellular machinery, orchestrating the creation of proteins that play crucial roles in the structure, function, and regulation of living organisms. So there are 20 different amino acids. Sequence of amino acids determines the type of proteins that will be made, and function of protein depends on structure. Next slide, please. So the genetic code, sets of three nucleotides in an mRNA strand that code for amino acid sequence in a protein. So the set of three nucleotides in an mRNA strand are called codons. Each codon corresponds to a specific amino acid, forming the genetic code for protein synthesis. This triplet code serves as the instructions for the cellular machinery to assemble amino acids in the correct order, ultimately determining the sequence and structure of resulting protein. So every three nucleotides in mRNA is called a codon, and each codon codes for a specific amino acid. Next slide. So there are 64 codons. AUG is a start codon or methionine signals ribosomes to start translating mRNA molecule. So AUG is the most common start codon and it codes for the amino acid methionine in eukaryotes and formyl methionine in prokaryotes. During protein synthesis, the tRNA recognizes the start codon AUG with the help of some initiation factors and starts translation of mRNA. So the three stop codons are UAA, UAG, and UGA. The signal determination of protein synthesis during translation. So when a ribosome encounters one of these codons in the mRNA sequence, it does not recruit a corresponding amino acid, but rather triggers the release of the completed polypeptide chain from the ribosome. So this process marks the end of protein synthesis and ensures the accurate and timely termination of the growing protein. Next, please. So translation, the process of assembling proteins from information encoded in mRNA and happens on ribosomes. So the translation in protein synthesis is the process where the information of mRNA is used to assemble amino acids into a protein. It occurs on ribosomes with tRNA molecules bringing the correct amino acid based on the mRNA codons forming the protein sequence. So ribosomes attach to ER to make membrane proteins and proteins that will be exported. Ribosomes and cytoplasm make protein that are used within the cell. Next, please. So the translation, the shape of tRNA impacts function. Anticodons of tRNA are complementary to mRNA codons. Pairing of anticodons to tRNA with mRNA codons ensure that amino acids are added in the correct order. So, the steps of translation. So, translation, it is the second stage of protein synthesis, unfolds in a sequence of essential steps. So, first is the mRNA, ribosomal subunit and initiator tRNA assemble on the ribosome. Then the amino acids are added by one to the growing polypeptide chain, guided by mRNA codons and complementary tRNA anticodons. Next is the ribosome moves along the mRNA, aligning the next codon to tRNA binding and shifting the tRNA molecules, and next is when a stop codon is reached, protein synthesis concludes. Release factors facilitate the detachment of the completed protein from the ribosome. So these coordinated actions ensure the accurate and orderly construction of proteins within the cellular machinery. So naay video para mas masabtan ninyo.
Initiation of translation in prokaryotes. The small ribosomal subunit is separated from the large subunit with the help of two initiation factors, IF1 and IF3. This complex then binds to a purine-rich region, the Shine-Delgarno sequence, upstream of the AUG start codon on the mRNA. The Shine-Delgarno sequence is base paired to a complementary sequence on the 16S rRNA, a component of the small subunit. This alignment ensures that the start codon is in the right position within the ribosome. Another initiation factor, IF2, brings in the initiator tRNA charged with initiator amino acid N-formyl methionine. The large ribosomal subunit joins the complex and all initiation factors are released. The ribosome has three sites. The A site is the entry site for new tRNA charged with amino acid or aminoacyl tRNA. The P site is occupied by peptidyl tRNA, the tRNA that carries the growing polypeptide chain. The E site is the exit site for the tRNA after it is done delivering the amino acid. The initiator tRNA is positioned in the P site. Elongation. A new tRNA carrying an amino acid enters the A site of the ribosome. On the ribosome, the anticodon of the incoming tRNA is matched against the mRNA codon positioned in the A site. During this proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right aminoacyl tRNA enters the A site, a peptide bond is made between the two now adjacent amino acids. As the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, the ribosome moves one triplet forward on the mRNA. As a result, the empty tRNA is now in the E site and the peptidyl tRNA is in the P site. The A site is now unoccupied and is ready to accept a new tRNA. The cycle is repeated for each codon on the mRNA. Termination. Termination happens when one of the three stop codons is positioned in the A site. No tRNA can fit in the A site at that point as there are no tRNA that match that sequence. Instead, these codons are recognized by a protein, a release factor. Binding of the release factor catalyzes the cleavage of the bond between the polypeptide and the tRNA. The polypeptide is released from the ribosome. The ribosome is disassociated into subunits and is ready for a new round of translation.
So, maura day ng RNA and DNA. So, in a summary, nucleic acid metabolism involves the synthesis and degradation of DNA and RNA. So, during replication, DNA is duplicated, uh, ensuring the genetic continuity. Transcription transcribes the DNA into RNA. So while translation converts RNA into proteins, DNA repair mechanisms correct errors and damage. So degradation occurs through processes like transcriptional attenuation and mRNA decay. So overall, nucleic acid metabolism is vital for genetic information storage, expression, and maintenance. So moving on, we'll have uh, the Novo synthesis, which will be shown in this video.
Hey guys, in this lesson I'm going to talk to you guys about pyrimidine and purine breakdown. So this is a follow-up video on my pyrimidine and purine synthesis videos. In this video I'm going to talk to you guys about just what happens when our cells actually break down pyrimidines and purines, what are some of the byproducts, what are some of the effects to human health if these byproducts of pyrimidine and purine breakdown aren't dealt with or aren't excreted properly. And finally, I'll talk to you guys about some drugs that we can use to actually treat some of these problems. So just briefly, what happens to pyrimidines? How are they broken down? Now, as I mentioned before, cytosine and thymine are part of DNA, and once DNA is perhaps degraded, you get a release of cytosine and thymine. Now, as I mentioned before, cytosine is actually processed to uracil through a deamination reaction, and both uracil and thymine can actually be salvaged and repurposed in the pyrimidine salvage pathway, and I mentioned this in my pyrimidine synthesis video, so if you want more information on the salvage pathway, just check that video out. Now, what will happen to uracil and thymine? They'll both be acted on an enzyme known as dihydropyrimidine dehydrogenase, and what will happen is they become, uracil becomes dihydrouracil, and thymine becomes dihydrothymine. Now, this step actually requires NADPH. Now, this step is actually the rate-limiting step of pyrimidine breakdown. Now, the next step actually processes dihydrouracil to N-carbamoyl beta-alanine, and dihydrothymine actually gets processed to N-carbamoyl beta-amino isobutyrate. Now, there's another common enzyme that processes these products. It's called beta-uridopropianase. And it actually, what it'll do is it actually processes the N-carbamoyl form of these two products into beta-alanine for the uracil pathway, and then beta-amino isobutyrate from thymine. And now these will be actually processed further. Beta-amino isobutyrate will actually be processed into succinyl-CoA through a couple of steps, and beta-alanine will actually be processed into acetyl-CoA. So both of these products can actually be used in the TCA cycle. Now, some of the byproducts include ammonia and CO2. So again, as we mentioned before, ammonia is toxic. We have to get rid of it, and the body gets rid of it through the urea cycle. So just know, guys, that uracil, the breakdown product, leads to beta-alanine and then acetyl-CoA. Thymine leads to beta-amino isobutyrate and then succinyl-CoA. So for purine breakdown, again, the purines are adenine and guanine. And the main thing I want you guys to get or take away from this is that purine breakdown product is uric acid. Now, this is actually a toxic molecule, so the body has to somehow get rid of it and somehow excrete it properly. Now, the daily production of uric acid is estimated to be about 600 milligrams per liter, and it actually has a very low plasma solubility of 70 milligrams per liter. So uric acid enters the blood, it becomes sodium urate, and it goes to the kidneys and excreted as urine. So this process is critical to get rid of uric acid from the blood. Now, the purine breakdown pathway actually starts with AMP. So AMP can get processed to adenosine. AMP can actually be also processed to IMP as well via an enzyme known as AMP deaminase. And as you can see here, an ammonium ion is actually produced from this reaction. As well, adenosine can be processed to inosine via an enzyme known as adenosine deaminase. And again, an ammonium ion is produced. So again, we know ammonium, we need to get rid of that, and that's done through the urea cycle. IMP can actually be reprocessed into inosine, and then inosine can actually undergo another reaction to become hypoxanthine via the enzyme purine nucleoside phosphorylase, and in so doing, it actually releases a ribose 1-phosphate. So once we get hypoxanthine, hypoxanthine can actually be salvaged in the purine salvage pathway, or it can be further metabolized into xanthine via the enzyme xanthine oxidase, and in doing so, it produces a hydrogen peroxide. Now, xanthine can be further processed again by xanthine oxidase to uric acid, and again producing another hydrogen peroxide. Now, this is the critical enzyme where allopurinol, a drug commonly used for gout, is actually important. So allopurinol is an inhibitor of xanthine oxidase, so that means that we actually don't metabolize hypoxanthine to uric acid, so it actually reduces uric acid levels when you take allopurinol. So this is the mechanism by which allopurinol acts. Now, the other purine, GMP, we never talked about that yet, but that can be processed into guanosine, which can then be processed into guanine via purine nucleoside phosphorylase, and again, it releases a ribose 1-phosphate from the reaction. So once we get guanine, guanine can actually be salvaged by the purine salvage pathway as well, or it can also be further metabolized by the enzyme guanase, releasing another ammonium ion, which then xanthine can also again be acted upon by xanthine oxidase to give you uric acid. So both GMP and AMP both lead to the production of uric acid. So as you can see, this pathway is critical for proper metabolism of purines, and if there's ever a problem in this pathway, you can have dire effects. And now one of the problems that can occur is a deficiency or a mutation in the enzyme adenosine deaminase, or ADA, which is the cause of about 15% of something known as severe combined immunodeficiency, or SCID. So just be aware, guys, that there are can be problems in this pathway that can be critical to proper body health and homeostasis. Now, I mentioned the purine salvage pathway, and I mentioned this as well in the purine synthesis video, but I just wanted to touch base on this once again. So, as I mentioned before in the last slide, that hypoxanthine and guanine can be salvaged in this pathway. Again, both hypoxanthine and guanine salvaging requires PRPP. Hypoxanthine gets processed to IMP, guanine gets processed to GMP, and this requires the enzyme hypoxanthine guanine phosphoribosyl transferase, or HGPRT. Now, IMP can be processed into GMP or can be processed into AMP in the actually purine synthesis pathway. So that's this enzyme is critical for purine salvaging. Now, if there's ever a problem with this enzyme, it can cause some very dramatic health effects. Now, there's one genetic condition that actually causes a deficiency in this enzyme, and that is known as Lesch-Nyhan syndrome, which is a genetic condition that is X-linked, and it occurs in about 1 in 380,000 live births. And now, what happens is, because this enzyme is deficient, there's decreased salvaging, there's actually increased metabolism because you're having an increased level of hypoxanthine and guanine, and that leads to increased uric acid. So these patients often get hyperuricemia. And also, because this pathway is not utilizing PRPP, PRPP actually increases as well, and that actually increases uric acid production. So as you guys remember from purine synthesis video, PRPP is critical for uric acid production. And I just want to talk to you guys briefly about the condition known as gout. Now, gout is the deposition of sodium urate crystals in tissue. It's because sodium urate, or uric acid, has a very low solubility in plasma. Again, remember, it's 70 milligrams per liter, so anything more than that will actually cause a precipitating effect on sodium urate crystals, and then you can get them lodged into tissues, which can cause many of the symptoms that we see in gout. So anyways, guys, I'll talk about that in another video a bit more in detail, but I just wanted to just touch base on that right now. So anyways, guys, I hope you guys found this video helpful. That was a quick lesson on pyrimidine and purine break.
So that's it. Thank you for listening to our report. So don't forget to subscribe our channel and ring the notification bell. And please comment down below your insights about the topic. That would be all. Thank you.